Scott Brown's Otology, Audiovestibular Medicine, Chapter 46, Anatomy and Embryology of the External and Middle Ear, Development of the Human Ear. Development of the human ear, the outer and middle ears. The 9 months from implantation of the fertilized and dividing egg, the blastocyst, to birth is divided into three periods, namely pre embryonic, embryonic, and fetal. The pre embryonic stages last 21 days, and the embryonic stage 35 days, and the fetal stage is longest phase at 210 days. During the embryonic phase, there is rapid growth and differentiation of the ecto, meso, and endoderm, so that by the end of this period, all the major organ systems have been formed, and the late embryo has an external shape that is obviously human. In the fetal period, there are changes in the shape, size, and orientation of various structures as well as rapid overall growth, but no new tissue development. During growth, from the fertilized egg into the fully formed fetus, animals pass through phases that represent to form degree at least their evolutionary precursors. The phrase autogeny recapitulates phylogeny has been used to express this reflection of earlier forms. In mammals, a phase is reached during early embryonic life when the mesenchyme surrounding the primitive foregut and pharynx differentiates into a maxillary and mandibular swelling on each side of the midline, just above and below the buccopharyngeal membrane. This membrane then breaks down, and a space, which will later become both nasal and buccal cavities, is formed. Further down the embryo and in the mesenchyme surrounding the pharynx, five or six parallel thickenings develop as bands that surround the pharynx. These are the branchial arches, which are numbered 1 to 5 from the head to tail. They are formed anterior to the 40 to 43 paired somites that subsequently give rise to the trunk and limbs. On the external surface, a groove develops between the each branchial arch, and that is matched by a cleft or pouch on the inner pharyngeal surface. In each branchial arch, a bar of cartilage, a group of muscles, an associated artery, and a cranial nerve supplying these structures and their derivatives all develop. The cranial nerve is called the post traumatic nerve. In addition, a nerve from the arch lower down supplies the inner endodermal surface of the arch above and is pre traumatic nerve. The first arch has the trigeminal nerve 5, the second arch the facial 7, and the third arch the glossopharyngeal 9. These are the post traumatic nerves. The pre traumatic nerves are the chorda tympani from 7, running to the first arch 5, and Jacobson's nerve, the tympanic branch of 9, running to the second arch 7. Derivatives of the first and second branchial arch. The innervation of the lower arches and indeed their derivation are much less clear. In fish, the layers between the arches break down to form the gill clefts, but in mammals this does not occur, although grooves on the external surface of the embryo do not develop and for a very short time comes into contact with the ectoderm lining the pharynx. However, the mesoderm rapidly intervenes and develops into the normal adult structures. Occasionally, there is failure of the system when the various branchial arches defect can occur as sinuses, blinded tracts opening onto an epithelial surface as a cleft or groove fails to regress, or less commonly as fistulae, a tract running from one epithelial surface to another when the ectoendodermal junction breaks down. The first pharyngeal pouch on the inside expands due to the rapid growth of the surrounding mesenchyme, and after dragging in some of the second pouch endoderm, results in the formation of eustachian tube, middle ear, and mastoid antrum. In creating these large spaces, the neural structures are forced to take a convoluted path to stay within the mesenchyme and yet remain bound to their original arch structures and the derivatives. The facial nerve is a good example as it runs posteriorly from the geniculate ganglion, then inferiorly and then anteriorly in order to leave the skull. The endoderm of the slit like sac, that is the precursor of the middle ear, lies against the ectoderm of the first pharyngeal groove by the fourth week. The mesenchyme grows in between these two layers to form the middle layer of the future tympanic membrane. The underlying sac expands as it reaches the developing ossicles and labyrinth. The epithelium is draped over these structures and their associated muscles, tendons, and ligaments, so that a complex series of mucosal folds is formed. The future eustachian tube lumen and middle ear spaces are formed by 8 months gestation, and the epitympanum and mastoid antrum are developed by birth. A few mastoid hair cells may be present at birth, albeit filled with amniotic fluid. However, development of the mastoid hair cell system does not occur until after birth, with about 90% of hair cell formation being completed by the age of 6, with the remaining 10% taking place up to the age of 18. The ossicles develop from the outer ends of the first arch Meckel's and second arch Ricketts cartilage that lie above and below the first pharyngeal pouch. The process begins at 4 weeks, and adult shape, size, and ossification are present by 25 weeks. The muscles attached to the ossicles arise from the arches that give rise to the part of the ossicle to which the muscles attach. Thus, the tensor tympani is attached to the upper part of the handle of malleus, which is derived from the first arch and is therefore supplied by a branch of the fifth mandibular nerve. In a similar way, the stapedius muscle is supplied by seventh facial nerve, the chorda tympani, which is the pre-traumatic nerve, is the second arch that supplies the endodermal structures of the first arch, that is, stays to the anterior two-thirds of the tongue, and submandibular gland secretomotor fibers has to run through mesoderm since it cannot run through air. This mesoderm subsequently becomes the middle layer of the tympanic membrane and is physical connection between the first and second arches. Table 46.3, derivatives of the first and second branchial arches. Cartilage, post-traumatic nerve, pre-traumatic nerve, and artery. First arch derivatives: Meckel's, malleus, incus, mandible, anterior malleolar ligament, sphenomandibular ligament. Post-traumatic nerve, mandibular nerve 5, pre-traumatic nerve, chorda tympanic 7. Second arch derivatives: Ricketts, stapes suprastructure, styloid process, lesser corner of hyoid, stylohyoid ligament, facial nerve 7, pre-traumatic nerve, tympanic branch 9, artery is the stapedial artery. The second external ear canal develops from the first pharyngeal groove in a complex fashion. A complete description is beyond the scope of this chapter and may be sought by from Michaels and Socket. It is sufficient to say that the meatus deepens by proliferation of its ectoderm, that an anteriorly placed bud of the epithelial cells expands vertically to form the skin, which will cover the future tympanic membrane. This clump of cells then opens up as a slit to form the canal lumen and produce the pars tensa and deep external canal epithelium. These two types of skin both have migratory properties so that the ear canal becomes self cleansing. The external ear or pinna, auricle or pinna, develops from a series of small cartilaginous tubercles that surround the first pharyngeal groove. These enlarge and coalescence, although it seems that the majority of the auricle is derived from the second arch cartilages, and that the tragus is the only contribution from the first arch. The rudimentary pinna has formed by 60 days, although it apparently continues to grow throughout life. Failure of formation of the first and second branchial arch structures gives rise to problems which range from failure of adequate skin migration with the ear canal cholestoma, which are very rare, through bad ears, accessory auricles, preauricular sinuses, to complete failure of formation of the external ear canal and middle ear. There are also the rare first arch fistulae, coloral fistulae, which comprise a tract between the external ear canal and the skin of the cheek, with the fistula path passing through the parotid gland and frequently between the branches of the facial nerve. The external internal ear labyrinth. The internal ear initially develops independently of the middle and external ears, although the two become interconnected by the stapes superstructure, becoming attached to the stapes footplate, thereby giving continuity to the auditory pathway. The development of the labyrinth can also be thought of as the initial development of the generalized structure of the membranous labyrinth, followed by a period of encasement by the bony labyrinth and the production of further series of spaces within the bony shell that in turn becomes the perilymphatic spaces of the complete structure. These different activities are happening at different times in different parts of the labyrinth, so that the damage or derangements at specific times give rise to many peculiar and varied abnormalities. Within the first few days of the embryonic life, that is at about 22 to 23 days, ectodermal thickenings form on the side of the head end of the embryo close to that part of the developing neural tube and neural crest cells, which will later become the brain and brainstem and the cranial nerves, respectively. The ectodermal thickenings of the aortic nerves, respectively, aortic placoid, which deepens and then sinks between the surface as the sides close in to form an aortic pit, which eventually loses connection with the surface and forms the aortic autocyst. Associated with the autocyst is a cluster of neural crest cells that later become the separate facial geniculate, auditory spiral, and vestibular scarpas ganglion cells bodies. The autocyst then undergoes a series of spectacular changes which result in the full sized outline of the adult membranous labyrinth by 25 weeks of gestation. The semicircular canal starts at develop around 35 days as three flattened pouches that outgrow at right angles from each other from the utricle. At the center of each semicircular ridge, the opposing epithelial surface meet, fuse, and then coalescence to be replaced by mesoderm. The superior canal is the first to be fully formed by 36 weeks. As these developments are taking place, the cochlea starts to be formed. The saccule, which is separated from the utricle, starts to put out a single pouch like process that grows and then becomes a coil from base to apex to reach its full 2.5 coils by 25 weeks. Within the membranous labyrinth, the sensory cells of the three cristae, two maculae, and the organ of Corti begin to develop from areas of ectodermal specialization. This is encouraged either by the ingrowth of nerve endings from the cochleovestibular ganglion, which was originally outside the autocyst, or by the development of the sensory cells encourages neural ingrowth. Figure 46.20, development of autocyst. 7 somite 22 days, neural groove, aortic placoid. notochord, aortic pit, and aortic cyst 30 somite and 30 days. Development of the membranous labyrinth. Aortic vesicle, saccus endolymphaticus, fifth week, sixth week, 10th week, superior vertical canal, ductus endolymphaticus, crus commune, horizontal canal, ampulla of posterior vertical canal, cochlea, saccule, utricle. Development of communication channels pass through labyrinth. Internal auditory meatus channel, persisting channel in cartilage model around 7 and 8 nerves. Subarcuate fossa, persisting vascular channel. Vestibular aqueduct, fifth and sixth ossification centers fuse around the endolymphatic duct. Vestibular aqueduct, cochlear aqueduct, resorption of precartilage. Fossula antefenestrum, resorption of precartilage. Fossula postfenestrum, inconstant, resorption of cartilage. Oval window, aortic capsule becomes foot plate of stapes and annular ligament. Round window, persisting cartilage becomes round window niche and membrane. The organ of Corte starts developing as a single block of heaped up ectodermal cells at about 11 weeks. Within this mass develops inner and outer cells, hair cells, and then specialized supporting cells. The tunnel of Corte appears between the inner and outer pillar cells as cluster of stereocilia and a single kinocilium are developing on each hair cell. The cochlear kinocilium regresses, leaving the adult configuration of stereocilia and the spaces between the outer hair cell opening up as the supporting cell, the detritus cells. Change shape. Differentiation progresses from the base to apex so that at any one time various stages of development can be seen in appropriately prepared material. Epithelium close to the sensory regions develops into the specialized cell groups that maintain the ionic and electrical stability of the endolymph. These regions are the stria vascularis of the cochlear duct and the dark cell regions of the vestibular sensory epithelium. The Bony labyrinth. The mesenchyme enclosing the otocyst becomes chondrified to form the aortic capsule. As the membranous labyrinth expands, the aortic capsule remodels and, in places, undergoes de-differentiation to form fluid-filled spaces that eventually become perilymphatic spaces. This de-differentiation does not occur where the nerves enter the sensory cells region. Elsewhere, the perilymphatic spaces becomes continuous and a communication with the cerebrospinal fluid is formed by the development of the cochlear aqueduct, which runs to the posterior cranial fossa from the scala tympani in the base of the cochlea. Ossification of the cartilaginous aortic capsule begins in and around week 16 from a variable number of centers that finally fuse without leaving telltale suture lines. This dense bony mass is the petrous bone. Interestingly, this is frequently the last part of a whale to decompose and is sometimes the only remains of those eaten by sharks. There are certain channels that remain within the aortic capsule, with one of the most important being the oval window, where part of the aortic capsule becomes the stapes foot plate and the annular ligament, thereby allowing sound from the middle ear to enter the labyrinthine fluid. The temporal bone. Four separate elements eventually fuse to form the temporal bone, which one is one of the most complex and interesting parts of the bony anatomy of the body. These four parts are the petromastoid complex, squamous portion, and the tympanic portion and the styloid process. The petromastoid is derived from the petrous bone described above and the continuing growth of its outer layers, which form the root of the roof of the middle ear. The wall, lateral wall of the eustachian tube, and the floor of the middle ear, the canal of the facial nerve, and the petrous apex. The squamous bone develops in the mesenchyme and starts to ossify from a single center close to the roof of the zygoma as early as 8 weeks. The posterior inferior portion grows down behind the tympanic ring to form the lateral wall of the fetal mastodantrum. The tympanic bone also forms in mesenchyme but from several centers around the external meatus. These form the major part of the ring with a groove on the inner aspect that becomes the sulcus for the tympanic membrane. Even by late fetal life, the bony ear canal is unformed, and only after birth do anterior and posterior protubercles grow to form the floor of the canal. These crescentric swellings eventually fuse laterally, leaving a gap in the middle ear of the floor. This is a foramen of Husky, which is usually closed by adolescents. The tympanic bone fuses with the mastoid process of the petromastoid and with parts of the squamous and petrous bones as the tympanomastoid, tympanosquamous, and petrotympanic sutures. The petrotympanic suture has a canal for the cauda tympani nerve. The styloid develops from two centers at the cranial end of the second arch, receives cartilage. The part close to the base of the skull starts to ossify before birth, but the styloid process itself does not start to ossify until after birth, and the fusion of the two parts mainly may not occur until after puberty. Much of the growth of temporal bone occurs after birth with enlargement of the whole structure and major growth of the mastoid process so that the stylomastoid foramen, which was initially close to the surface, becomes buried by the development of the mastoid tip. At the end and the same time, there is generalized downward rotation of the petrous bone so that the tympanic membrane moves from being almost horizontal in the neonate to an angle of 55 with the horizontal in adult. With these changes, the floor of the middle cranial fossa flattens, and the relations of the geniculate ganglion, the semicircular canals, and the internal auditory meatus change so that the middle cranial fossa surgery in the baby is anatomically a different experience from that in the adolescents. Thank you.